Hello everyone. Thank you so much for giving me the opportunity to present some information to you today. I very much wish that I could be there in person, but this is the next best thing. So I hope that you're having a wonderful meeting and getting lots of good information, and I hope that you will also find what I'm about to share with you helpful. Just a brief introduction. My name is Monica Coenraads. I'm the co-founder and executive director of the Rett Syndrome Research Trust. Before that, I had started the Rett Syndrome Research Foundation. So I've been doing this kind of work for more than 16 years. I have a daughter, Chelsea, who has Rett syndrome. She will be 19 years old next week. And she was diagnosed at the age of 2, and I live and work in Connecticut. Okay, so let's get started. I want to tell you a little bit about the research that's taking place and where treatments might come from. And give you a little bit of background on each of the therapeutic strategies that we are pursuing. First of all, I thought it might be helpful to put into perspective where an organization like RSRT and the research that we fund, how that falls together with other research that is taking place. And typically you have, in most countries, governments, in the US it's the NIH, in the UK, it'll be the MRC, that fund a lot of the basic science that happens, basic discoveries, and fund a lot of the laboratories in their particular country. And then from that research, there may be certain hints and clues, certain breakthroughs that happen that suggest that perhaps a molecular pathway or a drug target could be developed into a treatment for our children. And so this is called early drug discovery. Preclinical development is what happens typically in animal models like mice or rats or sometimes larger mammals. And then we go into the realm of clinical development, which is clinical trials in people, and then finally approval of a drug that your doctor can actually write a prescription for and that you can buy at the pharmacy. So those are, broadly speaking, the various phases that we have to go through, from a discovery in the lab all the way to a medication that we can give our children. So the government typically funds the early research that's taking place in laboratories. And then industry, meaning pharmaceutical companies or biotech firms, usually handle the clinical development, sometimes a little bit of the preclinical development, all the way to approval. But that leaves a big funding gap in the middle, which oftentimes creates a huge problem because there isn't funding to take a breakthrough that happens in the laboratory and to do all the necessary work that has to happen before it goes into clinical development. And that is where the Rett Syndrome Research Trust operates. We want to make sure that no good ideas that are happening in the laboratory end up falling through the cracks and disappearing. So that's where we work. So what do we do? Well, obviously we fund science, and of course that means that we have to raise money. So we spend a decent amount of our effort fundraising and then taking that money and funding it and funding science. Now, everybody talks about Rett research, Rett research. There is no one generic Rett research, and not all research is created equal. So really figuring out how best to spend the money that is so difficult to raise is really very, very difficult. Research is very risky. And I think that trying to figure out where to invest the money is just as hard, if not more difficult, than fundraising. We don't just fund science. We also identify therapeutic areas that need to be pursued. So we really partner with the scientific community to identify areas of interest. So for example, one of the strategies that you're going to hear later about in my presentation is a strategy where we're trying to activate the silent MECP2 that's on the inactive X chromosome, and I'll discuss that in a bit. But this is a strategy that now has gotten quite a bit of interest, and that even industry is becoming interested in. But this is something really that RSRT started. We had the idea, we talked to scientists, we tried to incentivize them to submit proposals to us. We funded first one lab, then two, then three, and now there's multiple labs, and now they're working together. So this is really something that we pushed, that probably on their own the scientists would not have done, because it was a very high-risk area. The same thing applies to modifier genes, looking for mutations in other genes, not MECP2, and other genes that might make the impact of having an MECP2 mutation less severe. This is also an area that nobody was pursuing, that we identified as a high impact area and went out and looked for scientists to work in this field. So we don't just sit back and wait for proposals to come to us, but we actually help to decide what areas we should go into, and then go out and try to find the best people to work in these areas. We recruit scientists into the field. We develop collaborations among scientists. So we have, for example, a gene therapy consortium, people that were working independently and were actually competing with each other, who are now working together. We fund an MECP2 consortium of 3 laboratories that are trying to figure out what MECP2 does. Again, these were labs that were competing against each other. Adrian Bird, Michael Greenberg, Gail Mandel, who are now working collaboratively. We organize scientific meetings throughout the year. Sometimes they're meetings of, like a meeting that we're having this November, for example, which is quite broad and covering all of the areas that we're funding and all of our scientists. Sometimes they're very focused meetings on one particular topic, where we really want to go into a lot of detail about a particular topic. Sometimes we have meetings for industry. These are meetings that we typically don't advertise. It's typically invitation only when we want to get certain scientists in the room. And we hold them throughout the year. Sometimes they're one-day meetings, sometimes they're 3-day meetings. We're also a clearinghouse of information. So clinicians, for example, doctors, medical doctors who want to be put in touch with scientists, or scientists who want to be put in touch with clinicians, or certain families that have interesting genetic situations going on. So we're kind of the go-to person for that, and trying to put people in touch with each other. And we're also, especially in the last 2 years, interacting very heavily with industry as they're learning and becoming more and more interested in Rett syndrome. So this is a rather important slide, and it looks at the various treatment strategies that we're pursuing. And I think it's important to also think about the difference between cures and treatment. So for me, a cure is, there's a saying, or a commercial here in the States for a company that does cleanup work after a disaster, like a flood or a fire. And their tagline is, like it never even happened. And so when I think about a cure, it's either, in the best case scenario, like it never even happened, Rett syndrome is gone, or really significantly dramatic improvements in the symptoms. That's what I would put in what I would define as a cure. Treatment, on the other hand, is an improvement in a symptom or a subset of symptoms. And of course, we want to pursue both, because as parents, we all know that we'll take any improvement that we can possibly get. But I think it's important to manage expectations about what certain drugs might be able to achieve. They may improve a symptom or a subset of symptoms, or can we expect something more dramatic? And so these are the treatment strategies that we're pursuing. The approaches that I think are going to lead to a cure, which is dramatic improvement, are the ones that I think are going to target actually MECP2, which is the underlying problem. The underlying, the root of the problem of Rett syndrome are mutations in MECP2, which lead to a protein that isn't working properly. So how do we address MECP2? Well, we can do that in a variety of ways. And they're listed there, and I'm going to be discussing each of those. And then we have suppressor genes. These are other genes, not MECP2, that can lead to having less severe impact of an MECP2 mutations. And then we have something I call downstream targets. These will not lead to a cure, but they will hopefully lead to improvements of symptoms. And there's lots of downstream targets that have been identified with various drugs that exist in these areas, some of which are being tried now. So I'm going to start with the first one here, activating the silent MECP2. So some of you will know this already, but I'll just give a little bit of a background. Because we know that Rett syndrome is caused by mutations in MECP2, and because MECP2 is on the X chromosome, we have, every female has 2 X chromosomes in every cell, and one of them is shut down. So in a particular cell, you might have the X chromosome that has the healthy MECP2 that is on, and the mutated one that is off. And so that cell will be making normal MECP2 protein. And then in the neighboring cell, you might have the opposite situation, and that cell would be making the mutant MECP2, which is not working properly. So in every cell that has a mutated copy of MECP2, there is also a healthy MECP2 in that cell, but it is silent because the entire X chromosome is turned off. And so this approach is trying to look for drugs that can turn on the MECP2 gene that is shut down. We like this approach because, number one, as I mentioned before, it addresses the root of the problem, MECP2. We like it because we don't have to deliver the gene, it's already there. And we also like it because we don't have to worry about too much MECP2. So we know from the MECP2 duplication syndrome that having too much MECP2 is a problem. It also creates a neurological disease. And so for gene therapy, for example, we have to be very careful that as we're delivering MECP2, we don't deliver too much. And so dosage may be a problem in gene therapy, but in this approach, it would not be a problem because we're only turning on the one gene that's already in the cell. It's also an intriguing approach because we don't need to know the function of MECP2. Don't need to know what it does and how it functions to be able to turn it back on. So we are funding a number of labs that are doing this. In the beginning, they submitted proposals independently, individually to us. But as we started funding multiple labs that were doing this, we cultivated an environment of collaboration among them. And we have conference calls every couple of months. We meet in person once a year. And these labs are sharing information, sharing any hits they might have, meaning any compounds that seem to turn on MECP2. And it's been a really rewarding experience to see these labs working with each other and in essence speeding things up. We're saving time because these labs are talking to each other and sharing information. There are a couple of hits that look interesting, a couple of compounds that, at least in vitro, meaning in a petri dish in a laboratory, not in a mouse yet, seem to be turning on MECP2, but there is still a lot of work to be done to confirm and to make sure that all the labs are getting the same information independently. So there's still a lot of work to be done. And as I mentioned before, this is an approach that industry is interested in as well. Okay, so that's one approach. The other approach is gene therapy. And I'm going to show you this video that came from a paper that was published a couple of years ago in Gail Mandel's laboratory. And the mouse that's running around has received gene therapy, and the mouse that is not moving is a mouse with no MECP2. That mouse has Rett syndrome and isn't moving, but you can see the other mouse how well it's doing. So this suggests, in fact, that gene therapy might be a good approach to treat Rett syndrome. And Gene therapy for Rett syndrome is it is a challenge. It's not an easy thing to do. First of all, it's a diffuse brain disorder, so there isn't a particular part of the brain that we need to target. We need to target the entire brain. So you need to be able to deliver MECP2 to every part of the brain. As I mentioned earlier, there is a potential risk of delivering too much MECP2, and so we have to figure out how to avoid that. And the reason for this is that some of the cells have normal MECP2, and some of the cells do not have MECP2. So if we could figure out a way to potentially get into the cells that don't have MECP2 and avoid getting into the cells that do, that would be a big step forward. So how are we going about addressing some of these challenges? So as I mentioned, we are funding a gene therapy consortium. These are 4 individual labs who are now working collaboratively together to execute on the experiments that need to be done and to tackle some of these difficult questions. 2 of them are hardcore gene therapy labs, Steve Gray from the University of North Carolina in Chapel Hill, and Brian Casper from Cleveland, a Nationwide Children's Hospital in Cleveland. And 2 of them are Rett labs, Stuart Cobb from the University of Glasgow and Gail Mandel from Oregon Science and Health University in Oregon, Portland, Oregon. And they're working on various things. Number one, they're trying to optimize the vector that is used. So the vector is actually, you can think of it as a Trojan horse that you package the MECP2 gene into, and it delivers the gene into the cells. There's lots of different kinds of vectors. We're trying to find the best one that can get into the brain and cover lots of area in the brain and get into lots of cells. We're also, you cannot fit the entire MECP2 gene into the vector. Even your tiniest genes cannot fit in their entirety into a vector. And so you have to pick, you have to select the various parts of the gene that you think are the most important. And so these labs are trying to figure out and optimize what are the parts of the gene that work that we need to package. So they're optimizing that as well. They're also trying to optimize the delivery route. Do we go through the bloodstream? Do we go directly into the brain? Do we go through the spinal fluid? So lots of different ways that it can be delivered. So as you see, they have lots of experiments to do to try to figure out the best combination of vector, packaging, and delivery system to achieve the best efficacy, avoid toxicity, and hit the most number of cells. Okay. The other approach is modifier genes. So we know from lots of other diseases that our own unique genetic makeup can either protect us from disease or predispose us to disease. And I'll give you an example. So, for example, we know people that smoke heavily that develop lung cancer, and people that smoke heavily that do not get lung cancer. It's their own unique genetic makeup that is contributing to whether they get cancer or not. We also know people that have low cholesterol levels who eat not very good diets and who don't exercise, and yet their cholesterol stays low. What's protecting them? It's likely their own genetic makeup. So we know that there are children and adults with MECP2 mutations that should cause full-blown Rett symptoms, but that don't have full-blown Rett symptoms. They have very mild. These are children that maybe talk, can walk, can run, can hold a pencil, might be able to sing, and something is protecting them even though they have a full-blown MECP2 mutation. So we're funding 2 projects in this area to try to identify these modifier genes. One is in the lab of Monica Justice. She was at Baylor, Baylor College of Medicine in Houston for many years, and then about 2 years ago she moved to Toronto. She's at the Hospital for Sick Kids. She has been funded by RSRT since we started. She has a very large project with many, many tens of thousands of mice that is aimed at basically giving these mice mutations through a drug, and male mice mutations through a drug, and then mating them with female Rett mice, and then you observe their offspring very, very carefully, and a certain percentage of those offspring will have no MECP2, so they'll have Rett, but they will also have inherited a random mutation somewhere in their genome. And we're looking for mice that are in much better shape than your typical Rett mouse. And then Monica Justice goes back and sequences the genome of those mice and identifies what the genetic mutation is. And she is halfway through this project, a little more than halfway through, and she has identified several dozen modifiers. One of those, which she has published, is in the cholesterol pathway. It's a gene called squalene epoxidase, and it has led to the clinical trial that has just started in New York. Using a statin, a drug that lowers cholesterol levels. But she has lots more modifiers. By the time she's done, we may end up with 50 or so modifiers. And the interesting thing is that these modifiers are not randomly scattered throughout the genome, but they actually fall into specific pathways. And so that gives us a clue that these are really kind of interesting potential therapeutic targets. We're funding another project with Jeff Newell, who is a physician scientist. He was also at Baylor for many years, and he recently moved to the University of California in San Diego, and he runs a Rett clinic there. So he is funded to do sequencing of the exome, which is the portion of the genome that produces protein, in high-functioning individuals with MECP2 mutations that are very high functioning, that can do that have lots of skills, and also on the other end of the spectrum, kids that are very severely affected. And we're trying to see whether we might be able to find what in their genome is leading to either them being very high functioning or very severely affected. These are a list of the clinical trials that are ongoing at the moment or that have recently finished or that will be starting soon. So ongoing at the moment is the statin trial, lovastatin, that's taking place in New York. The IGF-1, which I'm sure everyone's heard about, in Boston. Fingolimod is a drug that's used to treat multiple sclerosis. It boosts BDNF levels, which is why it's potentially of interest in Rett. This is a trial that's going on in Basel, Switzerland. Dextromethorphan is a drug that targets the NMDA pathway. This is a trial that's going on in Maryland and Baltimore. Starting soon will be ketamine. Ketamine is a drug that was suggested by David Katz, who found that it restored balance in various parts of the mouse Rett brain that were overly excited or overly inhibited, and that it restored balance. And ketamine is an anesthetic drug that's given for procedures and dental procedures in people, and actually in animals too, for veterinary procedures. And this trial will be starting shortly in Cleveland, Ohio, and they're going to be testing every child will go into the hospital 4 times and will be given ketamine, and then they'll be tested to see how they react. They'll be testing 3 different doses plus a placebo. So one of those 4 times they will not get ketamine. So every child will also be a placebo, and we'll see what happens. It's just the first step to see if ketamine, in fact, whether we see any improvements. And then recently finished trials were Copaxone. This was done by Sasha Dukic in New York. It was 10 children. She did see some improvements. It was a small trial. She's submitting her manuscript for publication now, and we'll have to move to a bigger trial. The NNZ drug, which is a drug from the company Neuron, they've done a small trial of adults, 28 days. They're now trying to do a bigger trial as well. And Epi-743 is a drug that's used for mitochondrial disorders. This trial was done in Italy. The company is called Edison Pharmaceuticals, and they're also looking to do a bigger trial. Besides funding basic science and the various strategies that we've just went through, we're also starting to become more and more interested in clinical research. How do we design better trials? This is difficult in Rett, not only in Rett, but in lots of neurological diseases. This is not unique to Rett. But how do we design good trials, and how are the measurements? What symptoms are we going to look at? And what tools do we use to measure those symptoms? And are they sensitive enough to detect perhaps small changes? These are very difficult questions that we're trying to address by working closely with a number of physicians. The other thing we're trying to see is whether we can identify a biomarker. A biomarker is something that we might be able to test in blood or urine or spinal fluid, or perhaps through imaging, that tracks the severity of the disease and that could track improvement. So you could imagine how wonderful it would be if there was something in a blood test that we would know whether a clinical trial that was testing a medication was working or not. This would make it a lot easier. So we're also trying to figure out if we can identify any of these biomarkers. So I wanted to end by saying that a lot of people have the misconception that because Rett syndrome is rare, that industry, and when I say industry, I mean pharmaceutical companies and biotech companies, aren't interested. But in fact, that's not the case. Over the last 5 or 6 years, industry has moved more and more towards rare disease. And the reasons for that are multiple. First of all, the reason is that oftentimes the biology of rare disease is better understood than common diseases. So for example, we know the cause of Rett syndrome. We don't really know the cause of Alzheimer's or Parkinson's or autism or schizophrenia or multiple sclerosis. So that gives us an advantage. There are doing clinical trials, for example, is much cheaper in a rare disease. If you're doing a clinical trial for a drug that's going to be used by millions of people, for example, like a cholesterol drug, you have to do clinical trials that are enormous. Because you have to make sure that about things like side effects that might affect a certain percentage of the population, and to be sure that you capture those, you've got to do very big trials. In rare disease, the trials could be quite small, maybe 20, 30 people. And so it's much, much cheaper to do clinical trials. Also, in the US and in Europe, there are lots of advantages that the governments give companies that are pursuing diseases that are rare, in terms of tax credits and waiver of regulatory fees and longer exclusivity where they can be the only people that are selling a particular drug before it goes generic, and also faster approval times. There's also reduced marketing cost. You don't need big sales forces, companies to sell to smaller patient populations that are very well connected. And finally, you can charge more. So a drug that might cost $10 a pill for a common disease might be much, much more expensive for a rare disease. And so annual costs for drugs that are affecting rare disease can range from 100,000 to 500,000, and so there's a very strong business model that exists. And for example, companies like Genzyme and Alexion and Shire, many companies that have developed robust business models for drug development for rare disease. So there's quite a bit of interest in Rett syndrome now, and I hope to continue seeing this grow. So I'm not there to answer your questions, obviously, but you can find my contact information on the RSRT website. My email is Monica, MONICA, @rsrt.org. And please, I'm happy to answer your questions via email. I think the bottom line is I continue to be very optimistic for our children. I'm encouraged by the number of both cure and treatment approaches that we're pursuing, and by the progress that's being made, by the caliber of the scientists that are working on Rett, and by the interest on the part of the pharmaceutical industry. So as a parent, of course, the progress never moves fast enough. But when I'm more objective and I wear my executive director hat, I can see that there's been a tremendous amount of progress. I also want to say that I know that many of you might feel cut off from information, and you feel like if you could just get to the States, or that things would be better. And we don't have a magic treatment here in the States either. My daughter is very severe. There's nothing really to help her here either. But I will say that in terms of information, you have access to all of the same information that our families in the States have. Through our website, our blog, the YouTube channels. I know that there may be a language barrier and that you might have to find somebody to help translate, but maybe that's what your organization can do to help, is to maybe take a lot of the information that exists out there and to translate it for you. But I would encourage you to sign up to our newsletter through our website so that you can stay informed, and I'm here to help answer any question that you might have. Thank you very much for listening. I wish you and your families and your precious daughters the very, very best. Please know that it is your daughters and my daughter that motivates and inspires me each and every day. Thank you.